Hello, am I audible to everyone?
Yes,
Yes. So hi everybody. Good evening. Welcome to second tutorial session. So today we'll be discussing again for the week two classes discussion on experimental biotechnology. Some okay. So So every week Tuesday 6 to 8 p.m. will be for 2 hours we'll be discussing any problem you have faced during the attending the classes or during solving assignments, and so we'll be discussing assignments which are you guys have already done, like the week 2 assignment is due on tomorrow. So I'll be discussing those assignments on the next day. So for today we'll be discussing those basic concepts, whatever Sarah has gone through in the first class of electrophoresis part 1. So we'll be discussing on the first part of electrophoresis. If you have any doubt, feel free to let me know, and please comment below in the comment section of the Google Meet. And if you have any doubt regarding the previous class, that also you can let me know, and we'll be discussing. So yeah, so let's start with today's class. So I'll be sharing my screen. I hope my presentation slide is visible and I'm clearly audible to you. So, hello everyone. I am Ananya Chatterjee, a PhD student of IISc Bangalore, and I am your TA or teaching assistant for the NPTL course Experimental Biotechnology. The course duration is July to October 2023. The course registration is still on. You can register, and you can also register for the examination. So there will be a main exam and some quizzes are also there. You have to submit those assignments to get considered in the registration for the examination. So be on time with submitting the assignment. So the week 2 assignment is live, and the submission date is due on like tomorrow. So please go through and submit the assignments. So yesterday, like the previous day, we were discussing regarding buffers, regarding pH, basic lab protocols, basic safety protocols, and we discussed that we'll be starting electrophoresis. So basically in this course, whatever we'll be discussing will be some techniques which are very much important in daily lab basis. Uh, I'll be keep admitting people. So yeah, which will be very much important for your daily day-to-day lab work. Whatever you'll be end up doing project. This experiments will be very much essential for you to know the principles and also simultaneously the practically. So you need to understand the basic principle behind these experiments very clearly. Now, what is electrophoresis? So here you can see, I'll, yeah. So here you can see that electrophoresis means running in the electric field. Okay. So yeah, I think it's yeah. So electrophoresis literally means running in the electric field. So you will be applying some electric field, and some molecules will be running or will be going with the effect of electrophoresis. Please let me know if my voice is clearly like. Just comment down that if it's not okay or something, then I'll be understanding. So the charged molecule moves to their counter-charged electrodes, but electric field is removed before it reaches the electrode. So in the electrophoresis you have to apply some electric field. Then what will happen? There will be some charged molecule which will move to their counter-charged electrodes. Okay, so there will be two electrodes, like positive, negative. So what will happen? The positive charge molecule will be going towards a negative charge because of the electric field. But as soon as you stop the electric field, you have to stop the electric field so that it doesn't go completely out of the frame. So movement of charged species in an electric field gives differential mobility to the sample based on the charge and consequently resolve them. Okay, so what is happening? You are giving an electric field, charge spaces are moving, and they have some differential mobility based on their charge and also based on their size, because if one molecule is very big enough, like its molecular weight is high, then it won't be able to move that first, right? So and so by that it will be moving, and consecutively it will get resolved also. So movement of the charged particle is retarded with the addition of polymeric gel, so that at sufficient time is available for resolving the gel. So while during this, like due to this electric field, when these molecules are moving, so we are giving one like matrix which is a polymeric gel. Why we are giving? Because polymeric gel is porous. Okay, so it's like why polymeric? Because we are adding some substance like agarose or acrylamide which gets polymerized. Okay, those macromolecules get polymerized or get interlinked with each other, so that they are making a polymer and which are gel like texture. So they have some porosity also. So with the pore only, those macromolecules will be run through. But if so, that pore size also matters. If the pore size is very big, your macromolecule will run fast. If the pore size is very small, your macromolecule will face some friction. Okay, so will not get sufficient time to resolve or something. So you have to consider the polymeric gel porosity also. So the polymeric gel is inert, uncharged, and does not cause retardation by binding the molecule. So one important aspect of selecting those polymeric gel you have to consider is that whatever substance you are using, it has to be inert. That means it's inactive. It has to be uncharged and does not cause retardation by binding the molecule. So the polymeric gel should not bind to the macromolecule whichever you have loaded. Instead, it forms of pores of different size depending on the concentration of polymer, and sample pass through this pore and as a result their electrophoretic mobility is reduced. So what is happening? The polymer is not interacting with any molecule. Like for DNA gel you will add DNA, for protein gel you will add protein. So those protein will not interact with acrylamide. Okay, and for DNA will not interact with agarose. What will happen? That because of those agarose or polyacrylamide, there will be some pores, so those macromolecules will be going inside those pores. So whichever molecule or macromolecules are big enough, those will face some retardation, and whichever macromolecules are small, those will not face that much retardation. So higher molecular weight will take much time to finish the run, and lower molecular will take less time to finish the run. So this is the basic concept of electrophoresis. So why do we do electrophoresis? So whenever you are doing some experiment, now in a basic way, how I can make you understand that you have purified some protein, okay, and those proteins okay, and so you have purified, and those are not that much pure enough, so it has some other contamination protein. Okay, so how do you know that you have purified something, okay, from the bacterial cell or from the mammalian cell, whatever it is, you have a crude solution. So that solution consists of multiple of proteins. So how do you know that if maybe your protein is of 80 kilodalton, so how do you know that your protein is there? So you need to isolate, or you need to separate all the protein macromolecules, all the protein molecules present in the solution. So the first and forward thing you will do is HDS page. So we talked about HDS page the previous day also. So HDS, what they do, they will just make the all the proteins in a linear peptides, and then will give a negative charge linearly, so that all the proteins charge will be same. Now what will happen, all the proteins will be getting separated based on their molecular weight. So this is the principle. You will do now HDS page, and you will get to see multiple bands. So in those multiple bands, you will go. So for HDS page, you will get to see monomeric bands, because you are reducing it, you are denaturing it, you are heating it while you are making sample. So you will get to see in monomeric band. So that's how you will be able to separate proteins. And now for DNA gel or the agarose gel electrophoresis, what happens? Now whenever you are doing plasmid isolation, that is you are doing DNA isolation or genome, like genetic material isolation, you need to know that whether your particular band size or whether your particular base pair fragment of DNA is present or not, or RNA. So we will run a gel, we will run agarose gel. So in that matrix is agarose. Okay, so in there also those DNA will get separated based on their molecular weight in the sense how much base pair is there. Okay, so this is the basic principle. Now you can see that whenever a charged molecule is moving and in an external field, why external field? Because in the electrophoresis setup we will see afterwards. So what happens, that you are applying voltage, you are applying current, so that there is an external field you are putting on. So there are external field you are putting on. So maybe this is your charged molecule. Okay. Now the external field whichever you are putting on, this acts on the molecule in a positive direction or in a forward direction. So this is a force from the external electric field, which is F equals to Q into E. We know the principle is that force equals to the charge of the molecule and the electric field applied on the molecule. So force equals to Q into E. So this is acting there. But simultaneously the molecule has also some frictional force or viscosity, you can tell. So this will act backward. Okay, that is a retardation force, you can also tell. So this is the formula is F equals to, or the frictional force or the retardation force or the viscosity is 6 into pi into eta into r into v. So this is the movement of charged particle in a external field. So here is some equation which I was going through, is that suppose a charged particle has net charge Q and the external electric field is E. Then the force F responsible for giving electrophoretic mobility, F equals to Q into E. So the frictional forces F, which is opposing the movement of the charged particle, is as follows. So frictional force will be F equals to small f into V. So where small f is friction coefficient and the small v is velocity of the electrophoretic mobility. So the movement of a spherical through liquid medium of the viscosity eta, the friction coefficient small f is given by, we know that small f equals to 6 pi eta rv. So the place where f equals to capital F or qv, so what will happen, the qv, whatever we are getting from electric field, qv equals to 6 pi eta rv. So the electrophoretic mobility small v is given by, so you can calculate from here, is given by q divided by 6 pi eta r. So as we know that charge equals to Z into E. Z is a valency of your molecule and E is the electronic charge. So Z into E will be the total charge of the molecule. So then the electrophoretic mobility can be expressed as nu equals to Ze divided by 6 pi eta rv. Okay. Any doubt, anyone? Okay, we'll proceed. So hence electrophoretic mobility V is directly proportional to the charge and inversely proportional to the viscosity of the medium. Also the size and shape of the molecule. So what are you getting, that whenever you are loading one macromolecule in a gel, so that macromolecule's electrophoretic mobility, that means the ability to move in a in addition to a external electric field, is directly proportional to the charge, okay, and inversely proportional to the viscosity of the medium, size and shape of the molecule. So in the case of relative mobility, it is directly related to the charge per radius of the molecule, okay. It's directly related to the charge per radius. So whenever your macromolecule is so small, its electrophoretic mobility will be higher. Whenever your macromolecule is big, its electrophoretic mobility will be lower, okay. So for a globular protein, the radius of the molecule is related to the molecular mass of the macromolecule. So the relative mobility nu dash or V dash is as follows: V dash equals to charge per mass. So if you consider globular protein, so if the radius of the molecule is related to the molecular mass, so if the protein's radius is big, that means the molecular mass of the macromolecule is higher. If the protein's radius is less, that means the molecular mass of the macromolecule is low. So the relative mobility V dash is as follows: V dash equals to charge by mass. So electrophoretic techniques, different types of electrophoresis techniques are designed depending on whether it is carried out in the presence or absence of supporting media. So while you will be performing different electrophoretic techniques, different types of you can be able to use supporting media, or you won't be using supporting media. So based on that, there are two types. One is moving boundary electrophoresis. What is this? In this method, the electrophoresis is carried in solution without a supporting media. The sample is dissolved in the buffer and molecules move to their respective countercharge electrodes. Moving boundary electrophoresis is carried out in a U-shaped, So it's carried out in a U-shaped tube with platinum electrodes attached to the both end of the arms. At the respective ends, tube has refractometer to measure the change in refractive index of the buffer during electrophoresis due to presence of the molecule. Sample is loaded in the middle of the U tube, and then the apparatus is connected to the external power supply. Charged molecules move to the opposite electrode. As they pass through the refractometer, a change can be measured. So as a desirable molecule passes, sample can be taken out from the apparatus along with the buffer. Okay. So what is happening? There is a cathode which is negatively charged. There is an anode which is positively charged. So there are cations, A plus, A plus, B plus, A plus, B plus, C plus, and there are anions. So it will move accordingly to their charges, and then you will be able to see that in the refractometer whenever your desired molecule is passing by. Okay. So but there is some disadvantage of this moving boundary electrophoresis, which are the resolution of the technique is very low. Resolution means the ability to differentiate or the ability to distinguish between two different macromolecules, two different proteins, two different DNAs, two different kind of RNAs and all. So the resolution of the technique is very low due to the mixing of the sample as well as overlapping of the sample components. The electrophoresis technique is not good to separate and analyze a complex biological sample. Instead, it can be used to study the behavior of the molecule in an electric field. Okay. Now there are another kind of electrophoresis, which is zone electrophoresis. So what happens here? In this method, an inert polymeric supporting media is used between the two electrodes to separate and analyze the sample. Okay, so in this method what happens, the previous method was like moving boundary, so there was no polymeric supporting media, only in the buffer you were dissolving your macromolecules and it was passing by. But here what is happening, an inert polymeric supporting media is used between the electrodes to separate and analyze the sample. The supporting media used in zone electrophoresis are absorbent paper, gel of starch, agar, or polyacrylamide. The major advantage of presence of supporting media is that it minimizes mixing of the sample and immobilization of the molecule after electrophoresis. It makes the analysis and purification of the molecule from the gel much easier than the moving down boundary electrophoresis. The gel electrophoresis is the best example of zone electrophoresis. Okay, so we'll be discussing that. Now, example. So as an example, maybe you were working with you have grown bacterial cells. Now what will do, you have like the protein, you have transformed the cells with some plasmid which will produce proteins eventually. Now you are trying to analyze the cell lysate. Okay, so you have collected your bacterial cells, you harvested those cells, you have lysed those cells, and from the after lysing, whatever proteins will be inside the bacterial cell will be come out, and you want to see what is happening. Is my desired protein is there? So what we'll do, we'll analyze the crude cell lysate in SDS page and determine the molecular weight of unknown protein. Principle of the technique: in the discontinuous system, negatively charged detergent, sodium dodecyl sulfate, is used to denature protein and impart a constant negative charge by mass ratio. Okay, so what I was telling, that in the case of SDS page, we use this substrate which is a negatively charged detergent, sodium dodecyl sulfate. So what it does, it denatures your protein of interest, every protein denatures your protein, and it gives a constant negatively charge along your protein. So it denatures your protein like that and gives a negative charge along the length of the protein. So this will be applicable for any protein of any molecular weight and all. So it will giving the similar effect to every proteins. Now, so what is happening now, as a result of separation of the protein largely depends on size only. So this technique uses 3 different buffers. So then in this SDS based system, you so while doing experiment, you need to know whatever buffers you are using, why you are using every constituent, you need to know why you are using this, why the pH is that. Okay, so what is happening in SDS page, you are applying similar charge. Now the resolving will be dependent on your only molecular weight. Okay, so this technique uses 3 different buffers. One is running buffer, another one is stacking buffer, and another one is resolving gel buffer. So what is that? A running buffer means, when I will be explaining the process of SDS based system, you will be able to understand clearly. But for now, running buffer is, so you have a you have a matrix, right? So that matrix will be any kind of gel. So for SDS base, that means SDS polyacrylamide gel electrophoresis. So the matrix is polyacrylamide. When you will make the gel, that means the gel of polyacrylamide, you have to add some other substituents so that the gel gets jellified or solidified, and you can keep the system vertically. So in this system, what do we add? We add APS and TEMET to start the solidification in your gel or start the polymerization. So after doing this, we need to keep the system matrix vertically, and during running the gel, that means whenever you were connecting with the electrode, and so that time you need to have a buffer, right? So that the buffer will help you in the electrophoresis, in help you in flowing of the ions. So for that, buffer is called running buffer. You will also need a stacking gel buffer, which you will need while making the gel, while doing the polymerization. And you need to remember and you need to know that stacking gel buffer pH is 6.8. Okay. And the another gel buffer which you need is resolving gel buffer, that pH should be 8.8. So in the case of discontinuous gel system, concentrate the diluted protein sample into a narrow band, allows application of diluted protein sample. Okay. What is the mechanism of stacking protein sample in the continuous gel system? So stacking gel buffer is composed of tris HCl, okay, and its pH is 6.8. HDS, where as resolving gel buffer contains tris pH 8.8 and SDS, and the pore size is large compared to resolving gel. Okay, so mostly the stacking gel is around 4%, and resolving gel you can use 10%, 12%, 15%, even 8%. Okay, so you can understand that what is 4%, 8%. So 4% means 4% of polyacrylamide, 12% is 12% of polyacrylamide in that system, in the gel. So how much the percentage of polyacrylamide will increase? What will happen to the pore size? The pore size will be decreased because there will be lesser space. So the mobility of chloride ion present in the buffer is more than the protein in the sample, you need to know, and the glycine moves slower than the protein sample. Okay, and as a result, protein sample gets sandwiched between fast moving chloride ion and slow moving glycine. So due to high electrophoretic mobility, the protein molecules run fast and stack between leading and trailing ions. Once protein sample enters into the resolving gel, where resolving buffer is of pH 8.8, glycine ion does not stop the migration of protein, and protein molecules separate as per as their size or molecular weight. Okay. Now, vertical gel instrument. So we'll have a schematic diagram of vertical gel electrophoresis apparatus, and it has two buffer chamber. Okay, sorry. So it has two buffer chamber, upper chamber and a lower chamber. Okay. So in the electrophoretic chamber you can see, so this is the upper chamber, and upper chamber and the lower chamber. Lower chamber is of tank, so and the electrode chamber. So both chamber are fitted with the platinum electrodes connected to the external power supply from a power pack, which supplies a direct current or DC voltage. So in the electrophoresis system, this is the tank. Here you put your gel like this. So this is inner chamber, this is the outer chamber. So both the chamber are fitted with platinum electrode, okay, which are connected to the power supply. Which will be connected to the power supply. So the upper and lower tank filled with the running buffer is connected by the electrophoresis gel casted in between two glass plate. Now there are two glass plate like this. One will be the thick glass plate and another will be the shorter glass plate or the short plate. So sorry. So short plate is of the size is small, it's thinner than the large plate. So how it will be done, whenever you will be loading, like whenever you will be starting your gel, starting casting your gel, what you will do, you will clamp this large plate and short plate like this, so that there is some space, and you will be adding this kind of spacer here, or comb. You can tell comb you will put, and so your gel will be made in between this fat plate and the short plate. So in between these two plates there is a little bit thin gap where your gel will be made. Okay, so your gel will have some thickness. Now you will be clamp those two glass plates and you will start adding your buffers. So at first you will be adding your resolving buffer, because your resolving buffer will be at the down. Okay, the resolving buffer will be here, and up above that your stacking buffer will be. So you will be pouring resolving buffer, and then you will wait for some time. So during this while, as soon as you will pour the resolving buffer, to reduce the exposure of this buffer components, that APS, with the oxidizing environment or with the oxygen, you have to pour some organic solvent. So preferably people put isopropanol. So little bit of isopropanol you have to put above this resolving gel, and you will wait for some time for the gel to get solidified. Now once the gel gets solidified, you have to remove, or you have to pour down those isopropanol completely, and you will add your stacking buffer. So you have to use some pipettes, slowly you have to add it. Okay, so that you are not disturbing the layer, you are not forcing the resolving gel. And likewise, as soon as you add the stacking buffer, you have to put the comb in. So the comb, what it will do, so you are adding one gel, so the gel will eventually get solidified, but now it's liquid. So as soon as you are putting some comb, okay, what will happen, it will create some well like structure, so that those wells you will able to load your sample. Okay, so now what it tells, that the upper and lower tank filled with the running buffer is connected by the electrophoresis gel casted in between two glass plates, which are rectangular and notched. There are additional accessories needed for casting the polyacrylamide gel, such as comb to prepare different well, spacer, gel caster, etc. Okay, these you need. So here is an example or a schematic diagram of different components of vertical gel electrophoresis apparatus. This is gel cassette, this is the tank, this is electrode chamber, this is comb, this are the wires you will be helping to like adding it with the power supply. So this is a gel cassette where you will make the gel. So this is, you can see, so what you can see, this will be your resolving gel, and like above that will be your stacking gel. So it will help in stacking your proteins in between chloride ion and glycine. Then in resolving gel, your protein will move faster. Okay, and then what will happen, you will be able to resolve your proteins based on their molecular weight. Now this is the tank, so you have to put it in electrode chamber where your positive and negative supply is there. So you have to fill this tank fully, and you have to fill this like electrode chamber fully, and also this tank you have to fill, and then you will be connecting with the power supply. So there is, you have to run your gel in a constant voltage around 100 to 130 volt. Okay, now buffer and reagent for electrophoresis. So the different buffer and reagents with the purpose of vertical gel electrophoresis are TEMET, number 1 is TEMET, number 2 is ammonium persulfate, number 3 is tris ACl, number 4 is glycine, number 5 is bromophenol blue, number 6 Kumasi brilliant blue R250, number 7 sodium dodecyl sulfate, which is SDS, number 8 is acrylamide. So number 1 is TEMET, which the chemical name of the TEMET is N, N, N prime, N prime, tetramethylene diamine. So what it does, if you add TEMET while preparing the gel, it catalyzes the acrylamide polymerization. So before TEMET you have to add ammonium persulfate, APS. So it is an initiator for the acrylamide polymerization. So APS is an initiator for the acrylamide polymerization. So after APS you have to add TEMET, which will catalyze the acrylamide polymerization. So tris ACl, another component is tris ACl. It is a component of running and gel casting buffer. So it will maintain the buffer. So for the stacking gel, the buffer pH is 6.8, and for the resolving gel it's 8.8. So is another component, glycine. It is the component of running buffer. Okay. So and the bromophenol blue, it is the tracking dye to monitor the progress of gel electrophoresis. So whenever you are adding one sample, in the sample loading buffer, you are also adding bromophenol blue, so that the dye, whenever it will pass by, so you will be able to see that blue dye is coming down. So you will be able to track that your proteins or the gels are running and how much it has progressed. Number 6 is Kumasi brilliant blue R250. So it is used to stain the polyacrylamide gel. Once your SDS page gel has completely run, you have to stain your gel. So it would be stained using Kumasi brilliant blue, which is a dye which will bind with the proteins and make your protein visible. Another constituent is sodium dodecyl sulfate, or SDS. It is used to denature and probe negative charge to the protein. We have talked about it. Another substituent is acrylamide, which is monomeric unit used to prepare the gel. So this acrylamide are monomeric. So whenever you are adding acrylamide and bisacrylamide, so this is the 9 components is bisacrylamide, which is the cross linker for polymerization of acrylamide monomer to form gel. Okay. Now the main component of the SDS page is polyacrylamide. So for that, which is that acrylamide and bisacrylamide, which acts as a cross linker and makes your gel polymerized. Okay. Now the process of casting of the gel. So the acrylamide solution, a mixture of monomeric acrylamide and a bifunctional cross linker bisacrylamide, is mixed with the TEMET and APS, okay, which are polymerizing agent, and poured in between the glass plate fitted into the gel caster. So there will be a gel caster or clip, you can say. So you will be clipping your long plate or the fat plate with a short plate. You will be keeping like this, but as the thickness is higher and the short plate thickness is thinner and there is one groove, so there will be a slight gap or the thin gap of certain millimeter will be the size of your gel or the thickness of your gel. So you will be able to make your gel like that. Now you will be pouring and mixed onto it and the gel will get polymerized. So what is the mechanism of acrylamide polymerization? You need to know. So ammonium persulfate in the presence of TEMET forms oxygen free radicals. Okay. So whenever you are adding APS and simultaneously you have to add TEMET also, so it forms some oxygen free radicals and induces the polymerization of acrylamide monomer to form linear polymer. So these linear polymers are interconnected by the cross-linking with bisacrylamide monomer to form 3D mesh. Okay, linear polymers will be cross-linked with bisacrylamide and will form some 3D mesh like this. And the size of pore is controlled by the concentration of acrylamide and the amount of bisacrylamide in the gel. In a vertical gel electrophoresis system, we cast two types of gels, stacking gel and resolving gel. First, the resolving gel solution is prepared and poured in the gel cassette for polymerization. Okay. A thin layer of organic solvent such as butanol or isopropanol is layered to stop the entry of oxygen. Why? Because oxygen neutralizes the free radical and slow down the polymerization, okay, and make the top layer smooth. So if you understand practically, what you'll see, you'll be like you will be keeping your glass bed like this. You'll be adding your solutions. As soon as you'll be done with your resolving gel pour, you'll be adding little bit of, like, you'll top up the resolving gel buffer with little bit of isopropanol, so that, or any organic solvent, so that you were not letting oxygen molecules interact with your resolving gel, so that it will not neutralize the free radical which forms during the interaction of APS and TEM, and also make the top layer smooth. So after polymerization of the resolving gel, a stacking gel is poured, and as soon as you will be pouring the stacking gel, you will fit a comb. Okay, a comb is fitted into the gel for construction of different lengths of the sample. So this is the chemical reaction between acrylamide and bisacrylamide. So you can see, while acrylamide and bisacrylamide you have mixed, so in your gel, like 30% acrylamide gel, you have acrylamide and also bisacrylamide, and you will be adding temit and ammonium persulfate with it. Someone wants to get added. So whenever you are adding temit and ammonium persulfate with it, this acrylamide and NN bisacrylamide will start cross-linking with each other. So what we'll see, we'll start cross-linking with each other. So here you can see the chemical reactions happening between acrylamide and bisacrylamide. So this bisacrylamide will be helping in cross-linking two linear chains of acrylamide. Okay. Now after this polyacrylamide matrix will be formed, as soon as the polyacrylamide matrix will be formed, then that gel, like, it will form a 3D gel. If you just visualize like this, it will form a like this gel, like this polyacrylamide gel it will form. And during formation of this 3D gel, there will be this pore. This is a pore, this is a pore. So it will be based on the percentage of your acrylamide. So this will be the gel cassette. So now the whole system is that this is the gel cassette, this is the electrode chamber, this is the tank. Okay, and now what we'll do, you'll be having everything together, like the gel cassette, you will be fitting in the electrode chamber, so that the gel will be connected with the electrode, and for the running buffer you will add inside the electrode chamber also, you will add inside the tank also, so the whole system will be put inside the tank. After that you will add buffer. Okay, and then what you will do, you will add your sample. So for adding sample, what you can do, you can use thin like pipette, you can use pipette tips, thin pipette tips, and then add the samples. And then you will just connect those tubings, positive to positive, negative to negative, and start the run at certain voltage. So after running, you will be, after while you will run the gel, as you added like bromophenol blue, you will be able to see dye font, like this dye font. So as soon as your dye font reaches here, you will stop the run and you will take out your gel and proceed with staining. Okay, just let me know if you have any doubt. If you have any doubt, please let me know. So, do you have any doubt, anyone? Okay. So this is the whole system. So if you are in bachelors, you have not seen gel run, you can. So there are several videos in YouTube. You can see those videos, and in this course also, in this experimental biotechnology course also, you will be able to see some demonstration video by professor of or the lecturers of this course. So don't worry. Now we will be discussing about running of the gel. So the sample is prepared in the loading dye containing SDS, beta mercaptan ethanol in glycerol to denature the sample. So beta mercaptan ethanol and SDS will help in denaturing the sample, and presence of glycerol facilitates the loading of sample in the well. So whenever you are adding the sample into the well, the glycerol is heavy, right? So it will help in loading. It will help in the like, so that by density it will increase the density. So by density it will be going smoothly inside the well. So as the samples are filled vertically, there is a distance drift between the molecules at the top V's and at the bottom of the lane. So this problem is taken care once the sample runs through the stacking gel. So the pH of the stacking gel is 6.8, and at this pH, glycine is moving slowly in the front, whereas tris ACl is moving first. As a result, the sample gets sandwiched between glycine, tris, and gets stacked in the form of thin band. Okay. Now, as the sample enters into the resolving gel with a pH of 8.8, the glycine is now charged. It moves first, and now sample runs as per as their molecular weight. So due to SDS, they have equal negative charge. So that's why they will be running as per as their molecular weight. After tracking dye reaches to the bottom of the gel is taken out from the glass plate with the help of a spatula, or with the help of like your hand also. You can just smoothly take it out, and gel is stained with Kumasi brilliant blue dye. So the dye stains protein present in the gel. So the dye stains specifically to the protein whichever are present in the gel. Okay. So this is all the principle behind those running gel. So you are adding stacking gel to stack your protein so that all the proteins come in a same lane or in a one lane, and then from there the race of or the run of your protein will start. So whenever your protein has higher molecular weight, it will run slowly. Whenever your protein has lower molecular weight, it will run first, and then you will be able to distinguish, or you will be able to resolve. So potentials of uh, potentials of discontinuous page. One is number of disulfide bonds, okay, and the second is separating proteins based on size alone. Please let me know if you have any doubt. Okay. So, what is the potential of discontinuous page? So, you can compare reducing and non-reducing denaturing gels can be used to provide information related to the number of disulfide bonds present in the protein. Okay. So, you can compare reducing and non-reducing denaturing gels, and you can provide information related to the number of disulfide bonds present in the protein. So if you know that your protein has disulfide bonds, and if you break those disulfide bond, then you will get this much of monomer. So then you can add BME. If you add BME, this will reduce disulfide bonds and you will get fragmented protein. And if you don't add BME, then disulfide bonds won't be broken. Okay, so this potential you have. Now, separating proteins based on size alone. In the presence of SDS and reducing environment, page gel resolve two proteins on the basis of molecular masses and the concentration of and the molecular masses and the concentration of gel. Okay, so in SDS page, the relative mobility and the log molecular weight as given by, so relative mobility V dash equals to V0 into A minus log M by M A. So molecular weight of the protein can be determined by plotting relative molecular mobility, relative migration, which is RF, relative mobility, with the log molecular weight of standard protein. So RF equals to migration of protein from the lane divided by migration of tracking dye. Okay. So as you will be adding bromophenol blue, so you will be tracking the migration of the dye also, and you will have a migration of protein from the lane. So this is the relative mobility. So let's see, this is the gel after staining and destaining. You have also loaded protein marker. So this is marker, this is sample. So you have to calculate the relative migration. So determination of molecular weight using SDS page. So this is SDS page and this is the determination of molecular weight. So this is D1, D2, D3, D4, D5. These are mean distance traveled by marker protein. So these are mean distance traveled by marker protein. So relative mobility equals to mean distance traveled by protein divided by distance traveled by dye. Okay. Now if you plot the graph, so there will be log of molecular weight in x-axis, and in y-axis we will have mean RF. So and if you plot the graph, you will get that the mobility as per as your molecular weight. Okay, so this is how you can determine molecular weight using SDS page. So this is the calculation. Now calculation of molecular weight of the unknown protein sample is a 5-step process. Resolve the protein sample on the SDS page alone with the molecular weight markers. Then calculate the relative mobility using the following formula. Relative mobility RF equals to migration of protein from the lane migration of tracking dye. Plot log molecular mass versus relative mobility of the standards. Perform a linear regression using a calculator or using regression software such as Microsoft Excel. Use the linear regression equation y equals to mx plus c. This is a linear like line equation, a straight line equation, to estimate the mass of the unknown protein. Log molecular weight will be slope into, which is m, slope into mobility of the unknown protein plus y. Y means the intercept. So, troubleshooting the pattern of the protein bands on a polyacrylamide gel depends on several factors. As a result, several types of defects are observed. Now we will discuss few of these defects and responsible factors. Okay. So while you will run gel, you can see some artifacts or some problems associated with your running, and they have some solutions also. We have certain troubleshooting you can do if you face those issues. So what are those? Smiling gel, that is uneven heating of the gel causes differential migration of proteins, with outer lens moving slower and the median lens. Rapid heat transfer eliminates this defect and be achieved by filling the lower tank with buffer until sample height. Okay, until your sample is there, you have to fill the lower tank completely, so that there is like proper heat transfer happens in the throughout the lens. Okay, so in this case smiling can be happen, and you have to do this proper filling so that you can eliminate this fact. Now the second thing is that diffuse protein bands. Okay, you can also see diffuse protein bands. So what happens, diffuse pattern of the band appears on the page. Diffuse protein bands pattern can be corrected by increasing running current by some percentage, higher concentration of acrylamide. So if you get to see diffuse protein bands, or like diffuse means it's like it's getting diffused from the gel, then you can increase the concentration of acrylamide, or you can increase the running current. Another problem is vertical streaking. So vertical streaking of the protein bands appear due to overloading of your protein sample. It can be corrected by either reducing amount of protein sample or running the gel at lower current. Okay, protein runs faster than expected. In few cases, migration of protein is not proportional to the molecular weight. So protein runs faster than expected. So what happens that in few cases migration of protein is not proportional to the molecular weight. It's either more or less on gel than the expected place. It is due to unusual very high proportion of basic or acidic amino acid. So then it can be happen that your proteins are running faster, it's not running as per as your marker and moving. So it can be happen if your protein has very high proportion of basic or acidic amino acid. Double bands. Appearance of double bands is due to partial oxidation or degradation of the protein sample. Addition of more amount of reducing agent or preparing a fresh sample will reduce these artifacts. Addition of more amount of reducing agent or preparing a fresh sample will reduce these artifacts. Or distorted protein bands. So appearance of distorted or uneven protein bands is due to stacking gel polymerization. It can be corrected by increasing amount of ammonium persulfate and TEMET or denaturing the stacking gel. Lateral spreading, it can be happen if protein bands appears laterally spreading. It can be avoided by reducing the time between loading of the sample and running. So sometimes you are loading your sample and you are waiting for some time. So what can be happened is that your proteins start diffuse. Okay. So don't do this. You have to reduce the time between you are loading and you are starting running the sample. Then we'll be discussing with the with the staining of page gels. So other variants of page Native page, SGS page discussed in the previous lecture. So in the previous slides, you can see that those are anionic detergent using sodium dodecyl sulfate and beta mercaptan ethanol to give equal charge to all protein and breaks the disulfide linkage. As a result, the 3D structure of the protein is destroyed and it migrate as per as their subunit molecular weight. So in the native page, the sample is prepared in the loading dye, does not contain detergent or any denaturing agent, and as a result, sample runs on the basis of charge per mass. In native page, the 3D conformation as well as activity of the protein remains unaffected. In urea page, in this method, insoluble protein is dissolved in urea and samples separate based on their charge per subunit mass. A gradient urea page is used to monitor the folding states of a protein. Staining methods in polyacrylamide gel. So there are different staining protocols are developed to stain the protein bands in the gel. Few of the selected staining protocol are given. So we'll be seeing and we'll be also discussing those protocols in later verse. In the next class also we'll be discussing uh, again electrophoresis part 2. So these are some different stains available for detection of proteins in polyacrylamide gels. One is Kumasi Brilliant Blue CBB, colloidal solution, silver stain, cypro ruby, cypro orange, cypro tangerine. So CBB feature is non-specific, one-step simple staining procedure. So non-specific means CBB binds to any protein. If you have protein in your gel, it will get bind to the protein, and it's one-step simple staining process means you will take out your gel after it runs, then you will add you will pour CBB solution, you will wait for some time, then once it stains properly, gets stained properly, you have to add this staining solution to remove the extra CBB bound to the gel matrix. And then detection limit of this CBB is 0.3 to 1 microgram per protein band. So if in one band you have 0.3 to 1 microgram, then you will be able to detect the protein. And then colloidal CBB solution, 5 to 10 fold more sensitive than CBB, no staining step is required. Silver stain, 100-fold more sensitive than CBB, no simple staining procedure, mass spectrometry incompatible. Okay. So once you do silver stain, it's very much sensitive. Okay, and it's not simple staining procedure, but it's not very complex also. You have to you have to have the protocol how you do that. We'll discuss in next class what is the principle of silver staining and how do you do that. But it's not mass spectrometry incompatible. That means you won't be able to cut the gel and give for mass spec. So the another type of stain is cypro ruby, mass spectrometry compatible, sensitivity up to 1 to 2 nanogram per protein band. Cypro orange, sensitivity up to 4 to 8 nanogram per band. Cypro tangerine, sensitivity up to 4 to 8 nanogram per band, can be used to detect blotted protein bands. Okay. So these are the different staining available for detection of proteins in polyacrylamide gel. So we'll be discussing in detail of the staining process, how do you do, and what are the principle basically, how CBB binds with the protein, what silver stain does, how we stain with silver staining, and so we'll be discussing. And in the next class, that is next week, Tuesday, from 6 to 8. And if you have any other doubt related to other class, you please let me know. We'll be discussing about that if you have any doubts. So I think you have gone through those assignments, and if you have any doubt, you can discuss. We can discuss, but we won't be able to discuss answers. But the basic concepts behind it, we can discuss. And after from the next class, I'll be starting discussing the assignments which are due on tomorrow. So I hope you have already going through the you are already going through the assignments and submitting. So if you have any other doubt, you can please write in the comment section. Please write down in the comment section. So specifically we'll be discussing the role. I don't know whether it's in assignment or quiz yet, but the gel mobility shift assay is basically what it does if you are doing electrophoretic mobility shift assay. So if you are adding your DNA and protein in, and if your protein has affinity towards DNA, then the DNA will migrate slower because already it is binding with something and the molecular weight is getting increased. So this is the principle behind EMSA, but in detail I'll be discussing in next class. I will point it down. We have not started covering gel mobility shift assay yet. Sorry, was I audible? I think my mic was switched off. So yeah, so the gel mobility shift assay we'll be discussing in the next class. So what happens in the mobility shift assay, you are adding simultaneously DNA or protein or two macromolecules which interact with each other, so that your the mobility or the mobility of your macromolecule which has affinity towards other macromolecules is getting retarded, or it's getting decreased. So that's why you will be able to get to see shift if you compare with the control. So and in detail the role of T4PNK I'll be discussing in the next class. If it's in assignment I won't be able to discuss today, but we'll be discussing in detail. And also the Kumasi Brilliant Blue, what it does specifically binds with proteins in your gel. If it so whenever you are running SDS page, so your proteins are moving, so the dye has affinity towards protein, so so that's why it gets bound within that portion of the gel which has protein. So that, so it's also one interacting, like the dye has an interactive affinity with the protein, so that's why it binds. But it's not very much specific, it will bind to every protein. It's not that it will bind to only your protein of interest, not like that. But yeah, and along with that the Kumasi dye also binds with the matrix, but we need to do destain to get rid of the non-specific binding of this stain with the gel matrix. So yeah, and the interaction, if you want to know the reaction between CBB and your protein, I will like which amino acid, how in the amino acid, which like with the which group of the amino acid the CBB is binding. I will have the reaction mechanism the next class. Any other question? Okay, so I'll be explaining again this Kumasi brilliant blue with the reaction mechanism in the next class. Uh, Hari Narayana, if you have any question, please write down. Anyone has any question? Okay, so the gel mobility shift assay we'll be discussing in the next class. So the principle behind is that if you are adding another substrate or another macromolecule with your macromolecule, and it's there is some interaction between that, so you can check that. So there will be some retardation if DNA protein interaction happens. So you will be able to analyze it in electrophoretic mobility shift assay. And the specific question you have asked, that what is the role of T4 PNK in gel mobility shift assay, so I have to check that. I will let you know in the next class. So as far as I know, that it's a kinase, so it will add ATP with your macromolecule. So the detailed role I will be getting you in the next class. Yeah, and what is the difference between T4 and T4 PNK? So so I'll be discussing in the next class. So PNK is the kinase and T4 is not. But what is the difference? Definitely I will discuss in the next class. If you have any additional doubt, you can let me know, which I'll be able to discuss next class. Thanks for your question, Harinarayan. So the next class will be starting from staining of page gels. Whatever staining I have discussed with you, CVB staining, silver staining, all these stainings mechanism I'll be discussing, and along with that I'll be starting discussing Western blot, which is very specific to your protein. We can use like primary antibody and secondary antibody. So I'll be discussing Western blot, and I'll be discussing agarose gel electrophoresis, and we'll be starting EMSA. And if you have any particular specific question like Harinarayana, you can just comment down, and I'll be get back with you with those specific questions. So your assignment deadline is 9th August, so you have to submit by tomorrow, and whatever you have faced problem we'll be discussing in the next class. And any specific doubt, please let me know so that we can discussing about those. And all the principle of Kumasi blue, Silverstein, like for the Kumasi blue, it interacts with protein, some specific amino acids like basic amino acids like lysine, arginine. So it interacts like with the interacts with the help of electrostatic interaction with those basic amino acids, with the positively charged basic amino acids. And also CBB has some it has hydrophobic interaction with the aromatic rings. So like that, Silverstein also precipitate proteins and like forms a, it makes the protein some like it's basically specific to cysteine amino acid of protein. So in detail we'll be discussing the Silverstein also in the next class. Any okay, what is native page and what is SE? So native page I previously discussed in the previous class. So native page means you will be able to see, or you will be able to visualize, or you will be able to resolve your protein as a native condition. So what is native? That you know the protein has primary, secondary, tertiary, quaternary structure. So native means whatever you, whenever you are isolating by purifying or getting the protein, the at the same state you will be able to visualize. You are not denaturing the protein, you are not reducing the protein. So like the protein you have, you will be loading like that with the loading buffer which doesn't have any SDS, which doesn't have any reducing agent. So while you will load, the protein will be get separated with the help of like charge per mass ratio, so that with the help of its own charge and own mass, it will get resolved. But in the case of HDS, what happens, HDS will give all the protein a linear negatively charged. So every protein's charge will be same, only the mass is the differentiating factor. So this is the difference between native and HDS page. In native page you will be get to know the exact molecular weight of a oligomer or a oligomeric protein. If your protein is hexameric, you will get to say the molecular weight of in a hexamer. If you but in SDS page you are denaturing the protein, so you will get to see the molecular weight of only monomer. So this is the difference. Any other question? I was not able to join the last session. Is there any alternative to cover them? So yeah, so we have a YouTube channel of this tutorials also. So you know that all the lectures from professors are getting uploaded in the YouTube channel of Nptel Experimental Biotechnology. And for my tutorial sessions, these are also you can see. So in the SWAM portal or in the NPTEL portal where your course links are, there is one link is called problem solving session. If you click the link, you will get to see there is a Google sheet of my with all the informations of my YouTube channel, of the presentation slides also you will get to access. So please go through the link and you will get to see the lectures. So the channel name is NPTEL Lectures by Ananya. Sorry. Yeah. So for two-dimensional EAG electrophoresis, I have not discussed today, but for brief overview, so you are dissolving your protein in two-dimensional. In one-dimensional means you are dissolving your protein with the basis of your molecular weight. But in the another dimensional, when you are doing it at 2D, what is happening? You are doing it another dimensional. Another dimensional, what parameter is there? The PI. It based on the PI. So it will have the gel will have a gradient of different pH. So where like which pH matches with the PI of your protein, there the protein will don't have any charge. So that's how the protein will get separated. So it will get separated by the molecular weight, as well as it will get separated by the pi. So this is the basic principle of 2D gel electrophoresis. So in this problem solving session will be basically, again I am telling for those who have joined new, so we'll be basically discussing whatever you were, like whatever sir is going through in these classes. If you have any doubt, please let me know. I'll be discussing. If I know the answer, I'll be telling you instantly in the class. If I don't know, I'll be get back to you with those answers. And regarding whatever assignments you will be giving, I'll be solving those problems with you. If you have faced any problem, I'll be discussing the assignment 1 or the mock assignment I already discussed the previous class, and the assignment 2nd I'll be discussing in the next class. So for that assignment the due date is tomorrow. So like this the class will go on. Does sodium dodecyl sulfate provide negative charge to electrophores? What do you mean by electrophore? So sodium dodecyl sulfate is a negatively charged detergent. So it denatures the protein, okay, and throughout it makes a protein denature. So the protein is not in a secondary structure now. The protein has become linear, so it gives the protein negatively charged. So HDS molecule will bind with the protein, so it gives the protein a negatively charged. So then every protein will have the same charge. So then the mobility will not be based on charge, but will be based on the molecular weight. Yes, it gives a negatively charged. Yeah, you can study native page, study protein interaction because if one protein is interacting with another protein and you are not denaturing those interaction, you are not hindering those interaction, if you load those, then you will get to see, suppose your protein is molecular weight of 40 kDa and the another protein is molecular weight of 60 kDa, so you will get to see 100 kDa band. Okay, so yeah, you can do that. So native means everything which are native you will be able to observe. Coercive reliant bulbu has affinity towards protein, specifically basic amino acids of protein, which has positively charged, that is lysine, arginine. So it has affinity towards those protein. It interacts electrostatically with those proteins, and with some aromatic amino acids it has some hydrophobic interaction. So yeah, it has affinity towards protein. So, Hari Narayana, whatever question you are asking, is this an assignment? Can you please tell? Because I'm not supposed to answer your assignment question. Sorry, so assignment question I am not supposed to discuss, but yeah, you could not find out in the lecture material. You please go through those available YouTube videos, and find it, because the assignment answer, we will tell you, will discuss you, but the thing is that, after the due date, not before the due date. So please go through those YouTube videos, and study from the book. You will be able to find in the wave also. You'll be able to find. I'll be able to discuss you on next Tuesday only. Can we rely on answers on wave on same topic? Look, you have to search through. You have to read through some lectures from books. You can go through some basic like biochemistry books, and experimental biotechnology, some techniques books. So where you can rely on answers, you have to rely on answers on good publications, or you can go through papers. So yeah, yeah. Okay, suggest other options. So options will be good books. You can go through some books like Lellinger's. Then there is another books uh, related to biotechniques. So whatever, like, books related to biotechniques, where you will be getting proper these techniques details like SGS, electrophoresis, those books please refer. Go through the lectures also, go through previous and the recent lectures. So if you know that there are previous lectures also available in the YouTube, so if you search through NPTEL Biotechnology, you will be able to get previous lectures. Assignment questions should be covered in the lecture material, but it can happen that the lecture material will be in the next week. So please go through the previous lectures also, previous year's lectures, so that you are a bit updated, and also go through books. But some answers you are not able to find, maybe in that specific lecture it's not there, but in the next lecture you can have. Like in the mock assignments, whichever was there, it was not covered in the week 0, but those answers was there, like is there in the week 1 lectures. So like that, if week 1 assignments you are not able to find, you please go through the YouTube channel of Experimental Biotechnology NPTEL, previous lectures. Maybe in week 2 you will be able to find it, and also simultaneously go through some books, so that you have some idea, and yeah, for answering assignments. And yeah, so protein can be measured in using nanorod. So you can measure the uh, absorbance of proteins at around 280, so nanometer wavelength. So you will be able to do that. No, you can rely on books, you can rely on NPTEL or any other courses. If you are like covering any specific topic, if you are learning about SGS page, please Google. Please, in the YouTube also there are very good lectures available. So along with NPTEL, along with different sources of books, you go through other lectures, other like platforms. So like education is open. Yeah, you can do that, but I am not supposed to tell the answer beforehand. That's the thing. You have to know that how, like, DNA is made up of, what are the molecules are there specifically, and how you can label those molecules, how you can like label some molecules which have some radioactive property also. So like this you have to understand and study. But yeah, don't worry about the assignments. Will those answers I will be discussing in the next class whenever the due date is over. But before that I'm not supposed to tell you the answers directly, but I can tell you that in the Lellingers or in some other books like standard biotechnology books, please go through and go through those chapters. In YouTube also there are very famous lectures available, and in the online also reliable platform please follow. If you are going through some research papers, there also will be getting sense. And in lectures I think whatever assignments are there, the question those should be covered in those are covered in the lecture. So please go through previous lectures also. If you don't know the YouTube's name, I'll just write you NPTEL experimental biotechnology, experimental biotechnology. This is the YouTube channel name. You'll be getting all those old videos. Those are 30 minutes 40 minutes video. Quickly you go through, you will get a sense. Online links for course. Okay, which course you were asking, Harinarayana? This course online link, online YouTube link. Okay, so you please search NPTEL experimental biotechnology. Whatever I have written, if you can't find, tell me, you will be able to find. In the YouTube, please search. There every lectures are there. Yeah, course name is experimental biotechnology. Sorry, the course name is experimental biotechnology. Yeah, so here what you can do, if you go to syllabus, not syllabus, so in assignments also you will get to see some modules. And yeah, so like here problem solving session, you can see those recording. If you want to book also, I think some, yeah, so there is one link of book. So this is the link of your course. I'm just I don't think so it's the link, but yeah, so this is the link. I think my screen is visible, so I am experimental biotechnology. Here there is a link, open link, so copy the link I have copied. Sorry. Okay, so please go through. No, there is no negative marking. If you don't have any further doubt, I'll be ending the session. we'll meet again. So assignments is accordingly like 35, 65 like this. So you have some certain percentage. It's written in the Nptel portal only where your course are. So it's how much I know it's 35, 65. So there is lesser weightage in the assignment, but its weightage is good enough. So if you don't have any further doubt, we'll be again coming back in the next Tuesday session with each assignment. There is no like you are having probably 100 like 10 questions. So if you see, you are having 10 questions in each assignment. So what's there like yeah, so every like all of the assignments you will be 10 questions, 10 or 15 questions, so you will be answering those. And then after cumulative marking, you will have 35% weightage for the assignment. Yeah, so that's all for today. We'll be discussing in the next class again. Please feel free to share any doubt, and next class we'll be discussing about electrophoresis in more detailed manner, and then we'll be discussing the assignments. What if someone forget to submit an assignment? Will the person be considered for exam? Person will be considered for the exam, but the assignment will not be marked. So yeah, I mean marks for each questions in assignment. It's again I'm telling you, it is a cumulative studies. Like if you are given 7-8 assignments, so if you are considering like 15 marks, so then one, if you can consider 1 mark. So the if you consider the total, then 35% is the weightage for total assignments, and 65% is the annual, like the final. So it's like that, either 30-70 or 35-65 like this. So don't worry about the how much marks is there. How much ever marks is there, the percentage will be same. So that's all for today. Thank you for joining in. Please join every classes, every tutorial classes, go through those lectures, come up with questions, discuss here, and we'll be happy to discuss, we'll be happy to take questions. Thank you all. Bye. Good night. Thank you all for joining. Next class we'll see Tuesday from 6 to 8. Please join. So the class will be going to 8. Whenever you have like, please join after 6:30, whenever you be there. The class will be on till 8. Thank you so much.